Hello students. In today's class, we are going to learn about lactose operon. So, we'll first discuss about what are operons. So, operons are basically consist of a group of genes that are expressed together under the influence of a same promoter. They are present only in case of prokaryotes, and the transcriptional unit in operons consists of a promoter region. So this is the region where RNA polymerase will bind and will identify the proper strand for transcription. Then we have other sequences which are known as operators. So these are DNA sequences that basically act as an on-off switch and can control if the transcription of the downstream gene will take place or not. And then we have the structural genes which encode for the actual enzymes or proteins. So operons, they are also regulated by regulatory proteins, other regulatory proteins which are known as repressors or activators. So operons can again be so lactose operon specifically, it is an example of an inducible operon. So it means that it is usually in an off state and it requires a signal or some indication in order to be switched on. Such operons which are induced by some kind of a signal or which express the gene in presence of a signal are known as inducible operon. So the lactose operon contains genes that are required for utilization or catabolism of lactose. Coming to the structure of the lactose operon, we have the promoter elements where RNA polymerase will bind, followed by the operator sequences, which are bound by regulatory proteins known as repressors. And we have the three structural genes Z, Y, and A, which encode enzymes beta-galactosidase, beta-galactoside permease, and beta-galactoside transacetylase, respectively. So, beta-galactosidase is involved in splitting up of lactose molecules into glucose and galactose, while beta-galactoside permease forms a pump in the membrane that will import lactose into the cell, while the beta-galactoside transacetylase is involved in a detoxification process that will remove or detoxify certain toxic end products that are generated during the activity of enzyme beta-galactosidase. So the question is, when is the lactose operon going to be expressed? So suppose that if the bacteria prefers using glucose as the primary sugar, then how does it control the expression of other sugar operons such as the lactose operon? So this basically also means that lactose is the glucose, is the preferred sugar. So there is no point in making all the enzymes and proteins that are required for utilization of lactose if glucose is already available in the environment. Okay, since this is going to be a waste of energy for synthesizing the proteins unnecessarily because the enzymes of lactose operon are basically for bringing in lactose, and then after breakdown it is getting converted into glucose only. So if glucose is already there in the cell, then there is no need for investing energy in expressing lactoperon genes. So what does the cell does in case the cell has presence of enough presence of glucose in the environment and when lactose is absent in the environment. Okay. So we have a regulatory gene which is known as a LAC-I gene present upstream of the Lactoperon genes, and so transcription and translation of LAC-I genes results in formation of a repressor protein. This repressor protein can bind at the operator sequence, and when it is present at the operator sequence, RNA polymerase cannot carry out transcription process since it generates a steric hindrance, and hence you will have no RNA formation for the structural. However, if we have lactose present in the cell, then lactose can bind with the repressor molecule and inactivate or change the structure of the repressor protein such that it is no longer able to bind to the operator sequence. So if it is not bound, then it is not creating any structural hindrance to RNA polymerase, and hence RNA polymerase can proceed with the transcription process, leading to synthesis of messenger RNA for the structural. So in this way, the presence or absence of lactose is going to make the repressor protein inactive or active respectively, and in this way it can control the expression of lactoperon. The another regulatory protein that is involved here is an activator protein, which is known as a CAP protein, that stands for catabolite activator protein. It is also known as cyclic AMP binding protein since it interacts with cyclic AMP and leads to formation of a cyclic AMP CRP active protein. So the another name for CAP protein is also known as a CRP protein. Okay. So the glucose concentration can affect the activity of the CAP protein or the CRP protein by modulating or changing the amount of cyclic AMP availability. So if you have high glucose concentration in the cell, then the effective concentration of cyclic AMP will be low and vice versa. So the CAP cyclic AMP proteins can bind the LAC promoters and will induce transcription. So when this will happen? When the glucose concentrations are low or high. So we'll look at this in the next slide. How does it affect it? So glucose basically is the substrate for glycolysis. It will enter glycolysis, and the primary purpose of glycolysis is to generate energy in the form of ATP. Now for generating ATP, it makes use of the available adenosine monophosphate or AMP, converting it to ATP. This AMP is also a substrate for another enzyme named adenylate cyclase, which is involved in converting AMP into cyclic AMP. So when we have high levels of glucose present in the cell, we have glycolysis happening at a high rate, converting all the available AMP into ATP. So the effective cellular concentration of AMP decreases, that leads to less availability for adenylate cyclase, and hence less amount of cyclic AMP is synthesized. So this means that high glucose concentration will lead to low cyclic AMP concentration and vice versa. So when we have a condition that is high lactose concentration and low glucose concentration, at that time we are going to have high cyclic AMP activity and high level expression of the lactose operon gene. So let us look at this, how this will happen. So when lactose is present in high concentration, first of all, it is going to inactivate the repressor protein. So repressor is unable to bind to the operator sequences. So RNA polymerase can proceed and can carry out transcription. Now, the rate at which RNA polymerase can carry the transcription, whether medium level transcription or high level of transcription, will depend on the presence of the uh, CAP cyclic AMP complex bound to the DNA upstream of the promoter. Okay. So the CAP protein has binding site for cyclic AMP. This leads to formation of an active cyclic AMP CAP complex, which can bind to sequences upstream of RNA polymerase, upstream of the promoter region or RNA polymerase binding sites. Through direct protein-protein interaction with RNA polymerase, it can increase the effective concentration of RNA polymerase near the transcription start site, which leads to high rate of RNA polymerase binding and transcription of the downstream structural genes. Thus, in presence of low glucose concentration, because of high cyclic AMP activity and hence presence of active CAP CRP, active CAP cyclic AMP complex, we will have a binding of CAP cyclic AMP complex at sequences upstream of the promoters, recruiting RNA polymerase near the promoters at high rate, increasing the rate of transcription manifold. Thus, we will have high level expression in presence of uh, high lactose concentration and low glucose concentration within the cell. So that's it about lactose operon. Thank you very much, and kindly like, share and subscribe if you like the content of